Welcome to the Entomology Lab at KCCR. My name is Hilary Ansa, and I'm excited to give you a brief introduction into our work here. As part of the Infectious Disease Epidemiology Research Group, our lab focuses on studying mosquito vectors and their role in disease transmission, particularly malaria. What we do in this lab varies depending on the research question we want to address. Because yes, even mosquitoes keep us on our toes. The Entomology Lab at KCCR was established through the EDCTP-funded project ASAP, which assessed the efficacy of Artemether lumefantrine and atovaquone progranil in treating uncomplicated malaria in African children, and also assessing the potential blocking effect of this triple therapy. Currently, we are working on the MAMCO project, which explores how malaria and comorbidities can influence an individual's attraction by mosquitoes. Now, let's take you through a typical day in the entomology lab, where mosquitoes are both a study subject and, dare I say, our most demanding co-workers. We begin by collecting pupa from the larval bowls and transferring them into beakers or pupal cups, which are then placed into the adult cages. Every day, we feed the larvae with ground tetramin to support their growth. The adult mosquitoes also get their share of sugar solution, because even mosquitoes need their version of an energy drink. To sustain our colony, we perform blood feeding by offering rabbit to female mosquitoes as source of blood meal to stimulate egg production. Once they've had their fill, egg-laying platforms are prepared by placing damp cotton and filter paper in petri dishes. These are placed in the cages overnight to collect eggs. The next day, the egg dishes are removed, and observing the tiny collection of mosquito eggs proves that our blood feeding efforts paid off. Periodically, we screen for microsporidial contamination in the colony. To do this, we aspirate a few mosquitoes from each cage, freeze them to knock down, and with a stereo microscope, dissect the midguards for examination. Within the research group, we work closely with the field team responsible of participant recruitment at the various study communities and sample collection, and the clinical laboratory team who is responsible of primary sample processing, which includes microscopic examination. Once the samples are received at KCCR, blood smears are prepared and stained for the microscopic detection and quantification of malaria parasites in the parasitology lab. For the ASAP study, participants carrying gametocytes are selected to perform the direct membrane feeding assays, that is the DMFA, to assess whether these sexual parasites can be transmitted to mosquitoes. For DMFA, we feed the warm blood from the participant to the starved female mosquitoes, and we look at their infectivity after 7 days by dissecting their midguards and check for oocysts. For the same participants, we compare the infectivity before receiving treatment and after receiving treatment. This is key in identifying factors that influence infection in humans to their transmissibility to mosquito vectors. For the MAMCO study, we test the attractiveness of mosquitoes by pairing infected participants to non-infected participants through a quasi-experimental setup. The two participants are moved to the malaria sphere, a semi-field system that mimics natural conditions for malaria transmission research. At its core is a 3-chamber olfactometer, which helps us study mosquito host-seeking behavior. The olfactometer is connected to 2 bed tents where the participants rest. These tents are covered with bed nets and overlaid with black material to prevent light penetration, ensuring that mosquito orientation is guided primarily by odors rather than visual cues. With this setup, we are able to assess which group mosquitoes are more attracted to, either the co-infected, mono-infected, or non-infected individuals. This provides valuable insights into how co-infections and comorbidities may influence host-vector interactions and malaria transmission dynamics. All results are then documented through data entry, ensuring that every detail is accurately recorded for further analysis and reporting. Together, our entomology, field work, and the parasitology work provides a comprehensive picture of malaria transmission right from the parasite in the human host to the mosquito vector.